Assalamualaikum warahmatullahi wabarakatuh, and hello everyone. We are from Group 2, and today we are going to discuss on 3 primer pairs that we have chosen for each respective genes. But first, let us introduce ourselves first. So my name is Afif Shafi'i bin Muhammad Puzi, number metric 205784. Next.
My name is Eng Jiaming, metric number 206504.
My name is Ahmadinur Benzali and my metric number is... Hello, my name is Ahmadinur Benzali and my metric number is 2071.
All right, Akmal, next.
Hello guys, my name is Siti Nur Putri Najmiah binti Kamar Bahrain. My metric number is 205412.
Next.
Okay, hello guys. My name is Amin Shukran bin Sharizam. My metric number is 207612. Nice to meet you.
Hi everyone, my name is Putri Zari Sofia binti Harzat and my metric number is 207878.
Hi, my name is Nuraini binti Mokhtar with matrix number 205304.
Hello, my name is Syed Funuhan bin Mazir. My metric number is 208241.
All right, so everyone have introduced themselves. Valentine have some technical issue, but she will join us if she can. So as a group of 9, we have decided to split into 3 little groups, with each group analyze their genes from the previous research technique in molecular biology assignment. So the 3 person then will work together to search for the preferred primer pair. So I would like to pass to the next person to talk about the first genes.
Okay, thank you, Afif. So for our first group, we choose the Texadene syntax, also known as Texa45, like the name shown in the slide. In short, we call it TS. So first, how do we obtain this gene? First, of course, we Google search about the gene involved in taxol production of the Texas brevifolia. Then we obtain the gene name, which is ATX. Then we search for the FASTA format. After search for the FASTA format, we convert it into the gene sequence by using EMBOSS. So after that, we discuss together and choose the primer randomly and manually. And then we choose 2 primers and design 2 primers, which is in the yellow and blue color. So a little bit down, Afif. So the first primer is in yellow color, which is forward and reverse primer. So after we chosen the primer, we insert the restriction enzyme sequence, which is HOL. Then the length of primer is around 17, then plus another 5 bases from restriction enzyme. For the melting temperature, forward and reverse primer achieve optimum temperature, which is less than 60, and the differences is less than 5. And for the annealing temperature is also, the differences in annealing temperature between forward and primer also less than 5, and the GC content is optimum, which is more than 50%. For the stability, we found out that the forward and reverse primer are stable at its end with a lot of G and C at the end of the primer. After that, we check with the primer blast. Based on the result, we obtain that we can say that this pair of primer is quite suitable because it has low self-complementarity value, and then the melting temperature and GC content also optimum. Okay, we proceed to the next one, which is in blue color. So for this one, we follow the step like just now. And then for the melting temperature, annealing temperature, and the percentage of GC content are optimum as well. And the stability, we found out that it's also stable because it ends with a lot of G and C. But after we run with the primer blast, down a bit, Afif, we found out that the self-complementarity value is quite high, which is 7 and 6. So after comparing these two primer, we choose Afif because based on the result of primer blast, the product of the primer also found that it is belong to the taxa species that we want. So next, I will pass to the next group.
Okay, so thank you. For the gene 2, we have decided to use DBAT gene, which are 10-deacetyl-baccatin 3, 10-O-acetyltransferase. So what we do is we find the FASTA format, which as you can see in the slide presentation. And then can you scroll a bit down, Afif? So this is the full length sequence after we convert it. And then can you scroll down a bit, please? Okay, so for my part is I just pick randomly nucleotide for my reverse and forward primers as I highlighted with the green color. And then like for forward primer, I just copy paste it. And for the reverse primer, I just reverse complementary it, which as you can see, the reverse primer. And then like the total length for the forward and reverse primer is about 17 nucleotide. So it is in optimum range. And then for the melting temperature for both forward and reverse primer, I just calculate it by using this kind of formula, which is 2 times 80 plus 4 times GC. So what I do is I calculate the 80 and the GC content, and then I just substitute it. And then for the both reverse and forward primers, I got 54 degrees Celsius. And for the annealing temperature is I just subtract it with 5 degrees Celsius. Then I got for both primers, which are forward primers and reverse primers are 49 degrees Celsius. For the GC content is I just calculate the GC and then like I divided by 70 and then I times 100. So both I got 58.82 percent. So the stability of 5' and 3' end, the forward and reverse primer are stable and as it ends with a lot of GC at the end of primer. So can you scroll down a bit, Afif? Okay, so as you can see that this is the result that I've that I just copy paste the full length sequence for my part. And then the result is I got 5 and 4 self complementary, but based on the result above, even though there are 4 and 5 self complementary, it can be used to design primers as it achieve optimum condition in temperature range, primer length, and GC content. So that's for my part, and I pass to the next presenter to continue this presentation. Thank you.
Okay, thank you, Akmal, for the brief information. So first of all, I would like to say thanks to the first group, which is Afif, Jamin, and Said, because we as a group 2 person, me myself, Akmal, and Putri, mostly of our result, we try to use your guys' result as our template. So thank you for the first. Thank you. And just like Akmal, so me and Akmal decided to do the primer pair together. So like Akmal also said that me also choose the forward primer and the reverse primer randomly from the sequence that we already converted by using the Emboss program. So for this primer, I choose 18 gene sequence and +5 from the RE from our lecture notes, thanks to Afif. And then for the forward primer, I get around 58 degrees Celsius. And for the reverse primer, I got 52 degrees Celsius for the melting temperature. So I think it's between optimum temperature. And for the ending temperature, yes, just as usual with -5 degrees Celsius. Okay, scroll down. So for the GC content, I got 61.11, and for the reverse primer, I got 44.44. But I think for the primer blast, I got a bit confused to use it. So I asked Akmal for help. And so I decided to compare my primer pair with Akmal or Kemal. So I see that the GC content for the forward primer is at highest, 72.22%. So I think the chosen primer pair might not be so optimized in order to amplify, since my self-complementary for the forward primer and reverse primer is 6 to 4. So it is higher compared to the pair chosen by Akmal. But then we decided to see Putri's result for the next presentation. Okay, let's move on to Putri's result. So yeah, Putri, you can try to give your opinion about your result.
Okay, thank you, Omi. So based on my result, I also take randomly primers from the gene sequence. So for the length of primer, I take around 18. And then for the melting temperature, I get for the forward primer, I get 66 degrees Celsius, and for the reverse primer, I get about 64 degrees Celsius. I think it is a bit high. It's a bit high because for the best range for the melting temperature is about is a range around range 52 until 58 degrees. So from my melting temperature, I get above 65 degrees and it have a tendency for secondary annealing. So then for the percentage of GC content, I get around, for the forward primer, I get 83.33%, and for the reverse primer, I get around 77.78%. So stability of 5' to 3 and 3' N, the forward and reverse primer unstable as it and with a lot of DNC of the end of primer. So can you scroll down? Okay. So I also use primer blast tool to get the predicted size. So I get around 662 base pairs. And both of the melting temperature discontent criteria in primer pair 1 have achieved minimum optimum condition. So I think I pass to Ami for the conclusion for our group.
Okay, for our group, we think that choosing Putri's primer is the best, as Putri's primer pair has a lower self-complementary than me and Akmal's primer pair. So yeah, that's from our group.
Okay, thank you, group 1 and group 2. So for the last genes that we have identified is Bacillin 3 amino phenylprophenol transferase, or in short, we just call it as BAPT. So based on this gene sequence, we have designed 2 sets of primer. And so for the first pair of primer, which is in green color, the length of the primer is 18 plus 5 from the RE insertion, so equals to 23 base pair. And the melting temperature for the forward primer is 54 degrees Celsius, And for the reverse primer is 56 degrees Celsius, in which both of the primer melting temperature is in between the acceptable range, which from 52 degrees Celsius to 58 degrees Celsius. It is important for the primer melting temperature to not be lower or even higher than the optimum temperature, because it will lower the efficiency of the primer and also can increase the chances of secondary annealing as respective. So for the annealing temperature for the forward primer is 49 degrees Celsius, and for the reverse primer is 51 degrees Celsius. And next, the GC content for the forward primer is only 50%, and the reverse primer is 55.56%, which is also in optimal range of primer GC content. And lastly, the stability of 5' and 3' end, in which the forward primer is more stable compared to the reverse primer, since it has more GC content at the end of the primer. Can you scroll down? Okay, so the figure here shows that the chosen primer was able to amplify the gene sequence by using the primer blast. And also the predicted size of the PCR product is 531 base pair. Next, I will pass to Nuraini to explain for the second set of primer.
Okay, thank you, Putri. So for my primer, I also choose randomly from the sequence. And the length of my primer is 17, which is the optimal length for a good primer. And after that, the melting temperature that I got for the forward primer is 54 degrees Celsius. And for the reverse primer is 52 degrees Celsius, which is we considered also optimal. And for the annealing temperature, my forward primer is 49 degrees Celsius and the reverse primer is 47 degrees Celsius. Also, both of them are lower than the melting temperature. And for the GC content, for my forward primer is 58.82% and reverse primer is 52.94%, which is also we consider is a optimal content for the GC. So both are considered as stable because both have a lot of G and C content at the end of the primers. So, okay. But after I put my primer at the primer blast, I found that my self-complementary is quite high, which is 9. And the expected product length is 399. So that's why we group 3 decided to choose Putri's primer as the most optimal primers because of the differences of the self-complementary, which is Putri's is more lower than mine. So yeah.
So to conclude, the gene that have been chosen, the design that have been chosen is from the group 1 is Afif, and group 2 is Putri Najmia, and also for the group 3 is the Putri one. So the reason why they have been chosen is because 3 of them have a lower value of self-complementary than the others. So I think that's all from us. Thank you.